In this video, we'll go over the answer key for Friday, April 10th. And our topic today is still membranes, but we're going to take a look at a much more zoomed out view of the membrane. And we're going to start with the Golgi. And the Golgi is made out of 3 to 6 distinct membranous sacs called cristae. And let's say we want a protein to go to the membrane. Well, they're usually going to go through the Golgi first. So remember how we talked about in the last video, the secretory pathway that goes to the ER. Well, once you're in the ER, you're going to end up in the Golgi. So when you go to the Golgi, you first start the cis and you'll exit through the trans. And during your travel through the Golgi, you're going to be modified in some manner. Now, there are different ways you can move through the Golgi. If you break off into little membranous sacs like this and travel from chamber to chamber, that's called antegrade transport. Or you could just chill in this chamber and the Golgi naturally moves up like a conveyor belt. So if you start in cis and just hang around in cis, eventually you'll make your way up the trans with what used to be called the cis. This is called cisternal progression. And it's actually possible to go backwards, called retrograde transport. And so the first problem is really just to test your understanding of what we just covered in that slide. So if you move in successive fashions, that's called antegrade transport. Now let's talk about vesicles. So when a protein leaves the Golgi, it does so in a vesicle. And let's say this protein needs to go to the lipid membrane layer. It will do that as a vesicle too. And this is how we can get proteins in the membrane. So remember in the secretory pathway, we said some proteins are infused in the membrane. So let's say we already have a protein that is infused in our vesicle right here. What can happen if we need it to be in the membrane is that it will come infused with the membrane and this vesicle will be part of the membrane. And now we have our protein part of the membrane. So that's one way we can start to embed proteins in the membrane. Now, vesicles, there are 3 known types of vesicles. We have Clathrin vesicles, and these things are just proteins, Clathrin, COP1, and COP2. Clathrin will take proteins from the Golgi, bring them to the plasma. COP1 will, what it will do is that any proteins that belong in the ER, that is, they have to stay in the ER, if they accidentally go to the Golgi, which could happen, they will be returned in COP1 vesicles. COP1 also goes by the name of codomer. COP2 will take proteins from the ER to the Golgi. So proteins can go to ER to Golgi, that's COP2. COP1 is the reverse of that, Clathrin to the membrane. COP2 can return any COP1 coded vesicles as well back to the ER. Or it will return via COP1 coded vesicles to the ER, rather. So we're going to look at Clathrin to begin. And Clathrin is made out of proteins called triskelions. It's this Y-shaped protein. And it's basically 3 heavy chains bound to light chains. And here is an actual microscopic image of that electron microscope. And it forms these cages from these shapes. And the reason why they're shaped like this is because you have a lot of different proteins you need the transport, so you want to make sure you have a lot of flexibility when it comes to how big you make the cage and what shape you make the cage. Normally, light chains will inhibit the heavy chains from forming the cage, so that's how you regulate this. And once the light chains are off, then the heavy chains can form that cage. Here is an image of COP1 and COP2. So KDEL says that you're supposed to be an ER protein. So most ER proteins in mammals on their C terminus, they have a K-del. So lysine, aspartic acid, glutamic acid, leucine. Yeast have H-del, or you can have KK, anything, anything, or KKK, anything, anything, anything. These C terminal tags say you're supposed to be in the ER. And let's say by accident that one of these proteins went to the Golgi. The way they go back is that we have specific receptors that bind to this sequence. And so they'll bind to this sequence, the protein, and a COP1 vesicle will form and then bring it back to the ER. So that is how we can return ER proteins back where they're supposed to be. So here is a question that gets at your understanding of our vesicles. So coated vesicles are used to ensure that enclosed proteins maintain proper orientation. Clathrin is one type. So what's true about Clathrin? What's true about Clathrin is that they transport proteins and GPI, so GPI is a lipid, linked proteins from the Golgi to the plasma membrane. Remember, that is what Clathrin is doing for us. And this again gets on the previous information we just covered. Which of these proteins are going to be ER proteins if we're looking at the C terminal tails? So that would be H-del, K-del, and KKEA, because if you just have the two Ks at the C terminal, that's good enough. All right, so one of the questions that scientists grappled with is, how do vesicles actually fuse? Because remember, on the outside of vesicles are charged phospholipids, and there's charged phospholipids here as well. How do you bring two things that are charged together? They're going to naturally want to repel if they're the same charge. And the way that we fuse a vesicle to the membrane is through these snare proteins. The vesicles have R snares, the membrane has Q snares. Estimated 5 to 10 snare complexes that will form. So what happens is that when you bring the vesicle and the membrane near each other, these snares will start to wrap around each other. And when they wrap around each other, they're forcing the vesicle and the membrane to fuse together. And somehow you have to get rid of these lipids. So these lipids are kind of just removed. Not 100% sure how that happens, but further fusion and fusion and fusion will happen until the vesicle is now part of the membrane, and whatever the vesicle was carrying, if this was a neurotransmitter, that is now released to the outside of the cell. And it's thanks to these snares that overcomes the penalty of bringing membrane heads together. Now, it's much more complex than these snares. However, we're not 100% sure how it all works, because experiments with just snares take about 45 minutes to fuse, while inside the body this happens in like not even a second. So there's unknown proteins that we're still not aware of, but this is the basics of how a vesicle and a membrane fuse. So question 4, which one correctly describes the usefulness of snares? That's 3 and 4. Post-translational processing that happens in the ER and the Golgi. We talked about the secretory pathway. Snares aren't part of that, but they help to overcome electrostatic repulsion, and they help anchoring between vessel molecules. We didn't really talk about that all too much, but snares can also do that. So some viruses have their own snare-like molecule. So here is the snare-like molecule for the flu, hemagglutinin, and this is what fuses the virus to our cells when they come and hijack our cell. So this protein binds salic acid. So salic acid is a type of lipid that your body will have. And what happens is when we bind that salic acid, this protein kind of flips out like a jackknife. So it's folded, but then once it binds this salic acid, it'll flip out and kind of stab kind of stab the membrane. It's bringing the virus and the membrane together, allowing the virus to put its genetic information into the cell. So that's how a virus or the flu recognizes what cell to attack and how to come close to that cell is through this hemagglutinin. And so this hemagglutinin is a great target if you want to try and inhibit this virus, because if the virus can't bind, then it can't make you sick. So question 5, researchers developing a new drug for the flu based on what we talked about the flu, which drugs are the best? All of the above. So if you can interrupt the conformational change in the pH, so let me just go back. When the pH changes, this can also make it flip. Drug that is responsible for cleavage carbohydrates on glycophorin A proteins. That's what the flu binds to. So if you chop those off, you should be good. Or if you bind hemagglutinin and you won't let it do its proper jackknife. Or rather, what this drug will do is that it will make the jackknife happen too early, and then your virus won't affect the cell. So D is the answer there. The last thing we're going to talk about is math, and what we need to know for math is this equation. And so we're talking about diffusion of ions or any substance. And the free energy of diffusion for any molecule, here's molecule A, equals RT, R is the gas constant, T is temperature, natural log of final concentration of molecule A, so wherever it's going to, divided by initial concentration of molecule A, that is wherever it's coming from, plus ZA. ZA is the charge of molecule A. So, for example, sodium is 1, chloride is -1, multiplied by Faraday's constant and the electrostatic potential. That is, what is the charge of where you ended up in minus the charge of where you came from. All right. So let's look at a problem that deals with this equation. So the sodium-potassium pump transports 2 potassium ions into the cell and 3 sodium ions out of the cell. If the membrane potential is -100, that is, the inside of the cell is -100 millivolts, the extracellular fluid has 140 millimolar sodium, 5 millimolar potassium, and the cytosol has 10 millimolar sodium, 100 millimolar potassium. What is the delta G for transport of 2 moles of K and 3 moles of sodium, and is this spontaneous? So for these problems, I always draw a picture. The circle is my cell. Inside my cell, I have 10 millimolar of sodium, 100 millimolar of potassium. Inside's -100. I'm transporting 3 sodium out, transporting 2 potassium in. Outside the cell, 140 millimolar sodium, 5 millimolar potassium. Alright, so to do this, we need to use the equation that we just talked about. Delta G equals RT ln final divided by initial plus ZAF delta membrane potential. So we have to do this equation for each ion. Let's do sodium first. So I'm going to ignore this 3, but I'll come back to it. So gas constant given 8.314. Temperature, we're at 310 Kelvin. So your gas constant's in Kelvin, make sure your temperature's in Kelvin. Multiplied for sodium, what's my final concentration I'm going to? So I'm going outside the cell, my final concentration is 140. What was my initial concentration, or what was the concentration that I came from? 10 millimolar sodium, so that's my initial. Plus the charge of sodium, Za, that's 1. Multiplied by Faraday's constant. Multiplied by my change in voltage. So the outside is 0 millivolts. How do I know that? Because the membrane potential of the cell tells you the difference between outside and inside. So if I say -100 is inside, and that's the difference between inside and outside, I can just say outside 0. my rule of thumb of this is to always make one side 0 if I can. It makes the math a lot easier. So delta charge is final minus initial, right? So my final charge is 0. My initial is negative 100. 0 minus 100 is 100 millivolts. You can see I have a 0.1 here. The reason why I had 0.1 is because Faraday's constant's in volts, not millivolts. So I converted this to volts. That's 0.1. All right, now let's get back to this 3. I have this 3 here because I need to do this calculation for every single sodium. So that's 3 sodiums. So I just multiply this by 3. I get 49351. Do the same thing for potassium. So see if you can follow along what I did with potassium here based on what I just said for sodium. I get negative 3854. This process is happening at the same time. So you add these together. You get a delta G of 45,496 joules. Now this is not spontaneous. Delta G, that's positive, non-spontaneous, negative, spontaneous, 0, equilibrium. And that's it for this problem set. If you have any questions about what I covered, please don't hesitate to ask. Otherwise, I'll see you in the next video.